Here.
Yes.
So good afternoon, Dr. Manikandan and Velayutham, and good afternoon to the participants who are watching this live session. And welcome everyone to today's session on unlocking nature's potential and peptide and therapeutics. This session is to embark on an exciting exploration in the world of peptides and nature's own biological building blocks and their emerging role in transformative medical treatments. And peptides is a short chains of amino acids have shown immense promise in addressing a myriad of health conditions and offering a blend of efficacy and specificity and safety that bridges the gap between the traditional and small molecule drugs and larger biologics. And today we will delve into the science of peptides. This highlight key advancements in peptide research and discuss the challenges and breakthroughs in developing peptide-based therapies and look ahead to the future possibilities that peptide the therapeutics holds. And join us as we explore how the careful unlocking of nature's potential through the peptides is paving new pathways in the realm of therapeutic development. Before we start Dr. Manikandan Velayutham session, I would like to give some short introduction about him. So we are privileged to welcome Dr. Manikandan Velayutham as our esteemed guest speaker today. So Dr. Velayutham stands out as an assistant professor and a dedicated researcher in the field of biotechnology with a tenure at the Institute of Biotechnology and Department of Medical Biotechnology and Integrative Physiology and Simmer School of Engineering and Savitha Institute of Medical and Technical Sciences, Chennai, initiated in December 2022. With a notable academic journey, Dr. Velayutham has held the positions from an assistant professor at Vyasya College affiliated to Periyar University, Salem, to a research scholar at the Department of Biotechnology and SRM Institute of Science and Technology, Kattanmulathur. His research has significantly contributed to microbial process technology and biotechnological advancements. So, Dr. Velayutham's educational background is impressive, holding a PhD in biotechnology and MSc from Bharatiya University, Coimbatore, and BSc from Vyasya College, Salem. His expertise spans academic teaching, administration, curriculum development, and pioneering research in zebrafish models and nanoparticle characterizations and toxicology and microbial diversity, focusing on groundbreaking drug discovery. His commitment and contributions to biotechnological research and education are well recognized and continue to inspire. Dr. Velayutham insights today promise to be invaluable and enriching our understanding of the potential within biotechnology. At the end, I would like to introduce to the motto of Dr. Manikandan, innovating for healthier futures through science. I would like to hand over the session to Dr. Manikandan and also thanks a lot for joining us today and over to you, sir.
So, very warm good afternoon to one and all present here, and thanks for a very nice introduction. So, can I share the slide now?
Yeah, yes, sir.
So, is it visible?
Yeah, yes.
Okay.
So just make a full screen.
Yes, I have given an full screen.
Yeah, okay.
Is it clear, everyone? Okay. So, yes, again, thanks for the opportunity, Times Tech Solutions and Mastering Up for giving opportunity to deliver a guest lecture on unlocking nature's potential peptide therapeutics. So overview of the presentation is about, we will be having a short introduction about what is peptide, what are the properties, and what are the different sources, and what are the different approaches for synthesizing the peptides, and what are the challenges, and what are the solutions, and some of the success stories, and future perspectives, how we have to focus on peptide therapeutics, then some our background story of how our research, which is towards correlating with the peptide-based researches, and take-home message. So this is all about the presentation that we are having. We are going through this. So in the day-to-day life, before getting into the regular topic. I'm just briefing out. So we are all having very various kinds of lifestyle, food style, and regular day-to-day life practices. And based on the natural environmental conditions and livelihood changes, day-to-day day life, we will be getting into numerous unrecognized diseases, and most of diseases still we are fighting for drugs. So drugs and drug-based therapeutics, it will be varies and based on different technological advancements. For example, stem cell-based therapies or molecular targeted based disease treatments, and some of molecules, compounds, in various aspects, the drug discovery research are emerging in today's life. So, in these all successive approaches, peptide-based drug discovery or peptide-based therapeutics is the one of the most significant advancement, especially in clinical research. So, what is peptides? We will know about DNA, RNA, and protein, amino acids, so on and so. So, what it is peptide? Peptide is if we simply says peptides are a fragment of proteins. So, we will know very well about it. Proteins which are made up of amino acids. Mixed, we have 20 different amino acids which will be classified into essential and non-essential amino acids, which will be regulating the protein synthesis, and it will be carrying out various cellular and immunological mechanisms and functions in our all the biological system. So all amino acids will be connecting together by with the peptide bond. So if the number of amino acids which is limited with 50 in numbers, that will be termed as peptide, a small fragment of peptide chain which is addition with amino acids. If it is more than 50, then it will be termed as a polypeptide. So if n number of polypeptide or mixture of particular number of polypeptide and particular amount of amino acids, then it will be, if it is in a larger number, that will be termed as protein. So in general, protein is a huge amount of amino acids which will be connected together in a polypeptide chain. But peptides are simple in structure. It will be limited numbers of amino acid. That is the amino acids which will be connecting with peptide bonds. So what will be the actual definition? So the definition of peptide: a peptide is the short chain of amino acids. The amino acids in peptide are connected one to one another in a sequence by bonds called peptide bonds. Typically, peptides are distinguished from proteins by their shorter length, although the cutoff number of amino acids for defining a peptide and protein can be arbitrary. So this will be the actual definition. So as I said, peptides is a short fragment of protein which will be having a limiting amino acids. So what are the different types of peptide? So these peptides can be classified into several types based on its physicochemical properties in nature. Then based on number of amino acids of combinations. Then based on different sources and based on different therapeutic applications. There are four major classes that will be classifying the peptides. So first one, based on the physico-chemical or structural configurations of the peptide, it will be classified into linear peptide, branched peptide, cyclic peptide, or semi-cyclic peptide. So these are the four different types of peptide classifications which can be classified based on the structural properties. So this will be the one type of classification of your peptide classifications. Then the next one is classification of peptide based on the composition or the constituents of amino acid. For example, dipeptide. Dipeptide is a peptide which is having two amino acids which will be connecting together with the peptide bond. If it is tripeptide, it will be three amino acids, followed as tetra, it will be four amino acids will be connecting together, and oligo, which will be more than five it will be termed as oligopeptide. If it is more than 10 or 20, more than 10, that will be termed as polypeptide. So based on the amino acid combinations or number of amino acids present in a chain, the peptide will be termed as di, tri, tetra, oligo, and polypeptide. So this will be the classification of peptide based on its number of amino acids present. So the next type of classification is what kind of source it is. So in the all over the globe, we will be having a n number of biological and non-biological living systems, such as microorganisms, then plants, animals. If we come to the animals and plants, again it will be a huge number of classifications like fungus, amphibians, and vertebrate, invertebrate. Then so different type of organisms, and if it is animals, will some animals will have a poisonous proteins also. So based on which source it is derived, it will be termed as plant peptide. Plant peptide, which means the peptide will be synthesized from plant-based products. For example, they will be taking an extract of a plant, whether it is a root extract or a shoot extract or leaf extract or floral extract, or sometime it will be vegetable extracts, or sometime it will be fruit extracts, or fruit peel extract, or food seed extract. So these are the different extracts that can be extracted from the plant, and that will be utilized for the peptide synthesis. Then again, microbial. So the microbial domain also will have huge number of classifications and n number of species. Based on the particular microbial isolates, it will be termed as microorganism based peptides. Then again, fungal resource, same invertebrate, amphibians, vertebrate, and venom. So all these are about source. Based on the source, we will be classifying peptide, naming the peptide based on its resources. Then one more, the next type of classification is potential bioactive functions of the peptide. So all the peptide which will be having a n number of amino acid and in combinations, and it will be having a various biological function. What are the biological function? For example, some peptide or some small molecules which will have a ability to kill microorganisms. So if it is the peptide which is killing the microorganism, that will be termed as antimicrobial peptide. Again, this antimicrobial peptide will have several classes like antibacterial and antifungal, and based on which type of mechanism it is targeting. Then, apart from antimicrobial, then we will be having antagonist peptide, neurobehavioral peptide, then um, what else? Neurobehavioral, anti-cancer, antiviral, and anti-inflammatory, then acetylcholinesterase peptide. So these are the different types of peptide which will be targeting based on the disease conditions or based on the biological function, antioxidant peptides. So in even our study, we have identified some antioxidant peptides which will be derived from the algal resource. So that will be termed as algal based peptides. Then again, which type of synthesis they are going to synthesis. Based on the type of synthesis, it will be having pure chemically synthesized peptide. Uh, Hello. So, based on complete chemically synthesized peptide and natural synthesized peptide and semi-synthetic peptide. So these are the different classifications of peptide. So I hope I will give a brief introduction about what is peptide, what are the classifications, everything. So, what is the drug status as of now? So, since from 1920 to 2020, so this diagrammatic representation will give a evolutionary prospects of the peptide-based drug development strategies. So, it will be a more than 20 years research statistics data. So, insulin is first synthesized or discovered medicine in the peptide history. So the second diagram, which will be representing the global market statistics reports, that the first figure, the global pharmaceutical market of 2019 report states that the peptide based marketing or the sales of peptide is more than 50 billion dollars in the global market. So the second diagrammatic representations, which represents how the drug approval gets evolved from 1960s to 2099. So until 2019, it is more than 52 drugs are approved as for therapeutic purposes. But right now we will be using more than 80 numbers of peptide-based drugs in therapeutic approaches. So again, the third C diagrammatic pie diagram, which represents the even the 5% of peptide drugs which will be having various therapeutic applications. So the peptide will act as an agonist 8% and inhibitors 9% and surfactant 2% and 80% of peptide drugs are against. Then therapeutic indications. So what are the various field how the peptides has been used in a therapeutic applications. So 11 percentage of peptides were being used in cardiovascular infections, 8 percentage of peptide drugs are used in reproductive medicine, 6 percentage in gastroenterology, and 4 percentage in osteology, and 20 percentage will be in different aspects, and 17 percentage of peptide drugs are endocrinology, and 17% metabolic indications, and 17 percentages of peptide drugs are oncological drugs. So this diagrammatic representations, which will be recognizing the evolutionary prospects of a peptide-based drug discoverable and marketable availabilities. So, what are the sources of drug? So, as I mentioned previously, 80 drugs, 80 peptide-based drugs are right now available in therapeutics for market, and more than 150 drugs are under the clinical research and developments. More than 600 plus number of peptide-based drugs are still in the preclinical trials, and n number of peptide molecules are in laboratory scale level, and most so many peptide based databases are available, and computer aided drug designing or computer aided peptide designing, which will be working more vigorously to identify or address the molecular target of upcoming disease conditions. So what are the sources of drug? So, as I mentioned previously, the peptides will have variety of resources. It can be isolated from plants, microbes, and animals in all environments of like landscape, marine environment, aquatic environment, and everything. So, so, so this is all about the sources of peptide which we can able to synthesize. So I'm moving to the next. So, what is the biotechnological approaches for peptide-based drug discovery? So, before getting into this particular diagram, I will be describing about the conventional method of peptide synthesis process. So, they will be having a two set of process that they will get a first they will make a extract so of a specific molecule, whether it is if it has a microbial resource or plant or animal resource, they will be getting a crude extract. That crude extract will be digested with a mixture of protein degrading enzymes. Then once the extract is processed with protein degrading enzymes, then it will be further processed with the help of SDS page and 2D or multitopsis technologies for separating and identifying the protein sequence fragments. Once the protein sequence fragments has been sequenced and identified, then it will be further processing of like in biological potentials. So this will be the conventional process, and it will be time taking also, because the extraction process may differs and it will have uh, so many optimization conditions. And again, preparation of extract and protein digestion and purification of protein, all this process will be time taken process. So what will be the alternate for the conventional method of enzymatic digestion of protein, or enzymatic digestion of a crude extract which will be obtained from the various resources? Then they will be going for chemical synthesis. So the chemical synthesis method is like a solid state synthesis method of the amino acids will be chemically added in one by one all together by so in a complete synthetic process. Then so this is also a time consuming process. Then what will be the advanced technology? So advanced technology is computer aided or in silico approach towards peptide designing or peptide predictions. So based on the recent evolvement of computer based drug modeling strategies, we can use with the n number of databases which will be available in NCBI or other proteomic databases, we will be getting n number of protein sequences in the databases. So with these databases and utilization and modern development of bioinformatic technologies such as like molecular docking, simulations, and ADMET property analysis, and various peptide calculator analysis. So these are the different softwares and tools which are right now available by easy accessible. So with these technologies, we can able to direct, or we can able to specify our what will be our drug target, what will be our protein resource, then how we can predict our sequences, then how the our peptide sequences which will target that particular specific disease causing proteins. So all the things we can able to done with the bioinformatic in silico methods, then it will be used for the further developmental status in the laboratory conditions. So here the schematic diagram which will be obtained are reported by Wang Li et al. In recent reports that applications of computer aided drug discovery therapeutic approaches towards the peptide based drug discovery. So the chemically synthesized peptides, which will be already reposited in a peptide database. So there will be n number of peptide data banks which are recently available in online resource. We can go through the uh, peptide libraries and we can screen and what are already reported peptide, what kind of literature data, or what kind of uh, recent researches which is already gone, or still it is n number of peptides are unexplored and unreported biological activities will be reposited in the peptide data bank. Then based on the peptide data bank literature survey and the informations, we will be either we can procure the peptide as by outsourcing, or we can synthesize our own peptide with the help of a proper chemist. Then we will go for further in vitro and in vivo screening of its biological potentials. So this diagrammatic illustration will also report the same, that the chemical peptide which will be deposited in the chemical library, sorry, peptide library. From the peptide library, based on this literature data, everything we will be either we will be synthesizing or we will be procuring the peptide by outsourcing. Then we will be starting in vitro, in vivo, and ADME in silico approaches to visualize the bioactive potential of a peptide, and we will be understanding what is the actual mechanism behind the peptide to target that particular biological functions. So these are all the recent approaches in basic drug discovery approaches on peptide based drug discoveries. So what will be the challenge? So why this peptide is always used
Sorry for
for?
the disturbance. I think slides are not moving, sir. I can able to see the fourth slide only now.
it visible now?
Now I think you are changing fifth slide, right?
Now I am in the sixth one. I'm
But
moving
I
to
can able to
seven.
see the fifth slide.
Now right now I am in the seventh
Ah,
slide.
yeah, yes, yes. Now okay.
Yeah.
Yeah,
Okay, I'm giving end to the slideshow because since it is in the slideshow, it is not moving. I think so.
yeah,
So
okay.
now it is visible, right?
Yeah, yes, sir.
Okay. Okay, I will go with this version. Is it fine?
Yeah, yes, sir. Okay.
Okay. So yes, we'll get back to the presentation. That all the drug discoveries have pros and cons. As well, the peptide-based drug discovery and peptide-based basic research also needs to be focused on various aspects. The peptides, which will be having hydrophobic, hydrophilic, and instability index, and net charge, and the pH, temperature, so many factors can influence peptide stabilities. So, what are the challenges? Why, since n number of peptide-based bioactive potentials are failing in the level of preclinical or clinical research. Why? What happening to these peptides? So, these are the critical conditions that peptide-based discoveries are. The researchers who is more intended to the peptide research, they must should be focused. So first one things they have to focus what will be the shelf life, which means the uh, half life of the peptide and what are the physical properties and what is the bioavailability at the time of administration and what is the structural sensitivity, whether the peptide can stable at pH or temperature or various enzymatic concentrations, the, then what will be its biodistribution or what will be the condition of the peptide when while it is getting into our gut microbiota system, because most of the peptides will have a shorter half life and there will be a most of the protein short amino acid segments are fragmenting or degrading in n number of factors like pH conditions, stability index, or what we can tell temperature and the enzymatic constituents. So it will make the active peptide into inactive stages. So these are the challenges that can be overcome by applying various techniques such as the modifications of peptide, like changing the side chain by increasing or reducing the side chain, or modifying the peptide secondary structures, or producing a D endomer or terminal capping as like a how to tell. Yes, this will be a very good example that like the post transcriptional modification, the protein maturation, the peptide also sometimes needs to be a specific target based delivery system. So either we can use nano encapsulation or nano emulsion or a specific or like a coating kind of thing that can specifically increase the shelf life or bioavailability or biodistribution ability of the peptide molecules. So extend like side chain modifications are enhancing the secondary structure of peptide by uh, changing the peptides with cross linkings or half life extensions, or identify possible molecular targets or structure in inducing probe. So with these are all some approaches and some more solutions still needs to be addressed, because all peptides having a unique properties and unique, so each and every peptide should be focused and specific attention needs to be given in order to maintain its stability and sustainability for its therapeutical approaches. It should not lose its bioactive potential, and it should not give any kind of side effects or toxicity at the time of usage, and it is also to be easy uh, for the route of administration. So these are the things that peptide-based research will be focused in a drug discovery aspects. So here. So in this slide, we will be having a list of peptide-based drugs which will be approved on from 2005 to 2020. So these are the as of now markedly available drugs for the various therapeutic purposes. So what will be the future direction of the study? So the future perspective of peptide-based researchers, which needs to be focused to study the to study should focus on investigation of the pharmacodynamic properties and understanding the functional mechanism of the peptide. And the bioactivity of the peptide will demonstrate higher in vivo models and clinical research, of course. Then the successful delivery mechanism of the peptide, because all the route of administration will vary, so that will be leads to the variations in the bioavailability. So we have to get a proper drug targeted delivery system for peptide based therapeutics. So those kind of approaches as like the functional mechanism addressing and bioactivity and stability increase all will cause prominent drug candidate of a peptide in future aspects. So this is an overview of what is peptide, what are different sources, how it is synthesized, what are the challenges, and what is existing therapeutic drugs. So now I am going to moving on other findings, which will be the work which will be done at the time of my PhD. I am going to give a very highlighted, very important results here. So, before getting into our peptide based discoveries, I'm just giving a background information about this peptide, but where it is. So, ours in my PhD days, my supervisor, Dr. A. Jesu Arogyaraj, he will be working on spirulina, which is Arthospira latensis. He wants to explore at the time of oxidative stress condition what will be the immunological response of the algal system. So he have introduced a hydrogen peroxide induced oxidative stress in the modified Zorut's medium for the spirulina cultivation. Why specifically hydrogen peroxide stress? Have you remember that? So after this COVID conditions, we are frequently sanitizing, we will be using some detergent. So in this case, hydrogen peroxide is a one particular chemical agent that will be most frequent combinations of our regular and sanitizer or detergents or wherever the cleaning agents we are using, there will be some proportion of hydrogen peroxide. So this hydrogen peroxide, this if it is prolonged use or overexposure, that will cause disease condition because it is a reducing agent. So, how this hydrogen peroxide and it will be correlating or it will be inducing the algal stress. So, this hydrogen peroxide with modified Zoruk's medium, which will induce sulfur deficiency conditions for Arthospira platensis growth. So, why this sulfur is essential for Arthospira platensis growth? Arthospira platensis will requires sulfur-based mechanism for its self nitrifying or balancing status of cellular chlorophyll content. So in the case, how it will be reacting. So this will be the study which will be performed in a transcriptomic approach. So in this transcriptomic approach, our lab had found n number of new antioxidant molecules which will be evolved in the transcriptomic database. So, ceryl O-acetyl transferase is the one of the protein which is expressed at the time stress conditions. So, this ceryl O-acetyl transferase protein, which will be regulating cysteine biosynthesis mechanisms in most of the microorganisms, but it will be reported from E. coli as well as salmonella in so and so. So this what is the role of serine versatile transferase in cysteine biosynthesis is this cysteine biosynthesis mechanism which will be converting inorganic sulfur into organic sulfur molecule in all microbe and plants. So it will be helping the serine O-acetyl transferase enzyme, which involves cysteine biosynthesis mechanism on algal cell of Arthospira platensis for the conversion of inorganic sulfur into organic sulfur for its reaction stage of hydrogen peroxide induced stress. So this particular protein will be selected for my study. We will be identifying a peptide which will be having 14 amino acid sequences. So in this diagram you can get uh, B is the proteomic structure of serine O-acetyl transferase. I hope you can able to see. So here the red highlighted portions is the peptide sequence. I have given the peptide sequence name and its chemical secondary chemical structure in the C diagram. Then we will be identifying, now we will be having uh, a peptide sequence. So all these analysis of identifying proteomic sequence and getting the peptide sequence, all this will be done with the help of bioinformatic analysis. So what will be our approach? So we will be having a transcriptomic data. From that data, we will be going through some series of bioinformatic applications tools for in order to identifying peptide sequences. Then we will be predicting the peptide sequence by active potentials in silico. Once we confirmation done with our confirmatory all properties, then we will be synthesizing the peptide in semi-static conditions by outsourcing. Then we will be screening its bioactive potential. So here the results which displayed here is the sequence and the helical wheel diagram. Helical wheel diagram is the identification of hydrophobic, hydrophilic, and other amino acid properties of the peptide sequences. So we predicted that the peptide which is named as NV14, which will be having 14 different amino acids, which will have the antioxidant potentials. So we will be further screening this peptide's antioxidant potential by performing uh, antioxidant various antioxidant activities such as nitric oxide and superoxide radical scavenging (ABTS), which means ABTS antioxidant activity, and hydroxide radical scavenging antioxidant activity assays. So our peptide, which will be shown a dose-dependent increased concentration of antioxidant potential, which will be compared with the standard antioxidant, which will be termed called Prolex. So then we will be evaluating the cytotoxicity profile of this NV14 peptide in MDCK cell lines. So we have used Triton X100 as a positive control and untreated will be kept as a control. Our peptide showed a similar percentage of cell viability. So which states that the peptide won't having any cytotoxic effects on MDCK cells. So since in the normal condition the cells the peptide won't affect any uh, cells. So we want to check what will be the antioxidant potency. So in order to evaluate the antioxidant potency, we have used MDCK cells in stress conditions. So we have developed the reactive oxygen species, which means the hydrogen peroxide induced stress model by the cells are treated with 200 micromolar of hydrogen peroxide with the culture medium with the absence of fetal bovine serum. So it will develop the stress conditions. So in this condition, how our peptide will be reacting. So this is what our objective to analyze. So C, control, it is the untreated, which means the cell is in the normal condition, and the positive control, which will be the cells are treated with hydrogen peroxide, and the other five images, which will be the cells which will be treating hydrogen peroxide treated medium as well our peptide. So the cells are treated for 24 hours, uh, 24 hours after we will be performing DCFTA staining. So DCFTA staining is the ROS, which means that ROS detection staining. So we can see that the positive control, which shows highest fluorescent intensity, and the peptide treated, which will be gradually decreasing its fluorescent intensity, and 45 micromolar of peptide, which also shows similar to the control. The same way, the next image, which is figure 10, which is the whole chest staining. Whole chest staining is a apoptosis staining. So, but the first image, which states that our peptide reducing the stress in dose dependent manner. So, it is reducing the ROH. So, as well as the second image, which will be performed as a Hodge staining, which states that how the peptide molecules, which is protecting the cell from stress induced apoptosis. So, the positive control, which is showing highest cell death rate, but the 45 micromolar of peptide treated cells, which is shown similar to the control group. So, the both the images, which will be confirming that the dose dependent increase in antioxidant potential and dose dependent decrease in ROS mediated cell death. So, which states that the peptide, which is having antioxidant potential. So we will be further screening its antioxidant potential on enzyme in the mechanism aspects in order to evaluate these three enzymes: superoxide dismutase, catalase, and glutathione peroxidase. So these are the major enzymes which is acting as a preliminary antioxidant pathways and antioxidant mechanisms which will be regulated in all these cells. So at the time of hydrogen peroxide treated cells, it will be having a reduced gene expression of superoxide dismutase, catalase, and GPX, whereas our peptide treated cells, which is increased and it is having a high fold of superoxide dismutase, catalase, and GPX enzyme regulations. So this states that the regulation of enzyme is getting high with our peptide treatment. So, with this, we will be telling that the peptide molecule which is having antioxidant potential. So, this will be our preliminary objective, and we will be further moving to the screening of anti-cancer potential of these MV14 peptide because it is having ROS balancing system. So, we will all know about that cancer. Cancer is the most curious disease which will be initiated due to the reactive oxygen species imbalance conditions. So, whatever the condition, either it is reduced ROS or increased ROS level, it will cause cancer. So, since it is balancing antioxidant system, whether it may work on anti-cancer, it will be our perspective. So we are initiated our anti-cancer screening on this NV14 peptide. So we have performed neutral red uptake assay and LDH assay. So we got dose dependent response against NCF cell line. We have performed with different cell lines for the anti-cancer profile for initial screening, but we get significant results on NCF7 cells. So we will be further getting micrographic images on and acridine orange and PI. So this will be a differential staining to in order to view the cells in which stage it is having. So we will be differentiating viable early and late apoptotic cells and necrotic cells. By this training, we will be getting early and late apoptotic stages of various cells in dose dependent manner. So we will be further screening its anti-cancer potential by fluorescence activated cell shortening. So that will be used for in which stage our peptide molecules are inhibiting this cell anti-cancer cells proliferation. So we are very well known about it. Cell cycle will have four stages, that G naught, G1 and S, and G2 M and division phase. So our peptide will act on G0 phase itself and it will be inhibiting the cell cancer cell proliferations of MCF7 at higher concentrations. Then we will be going what kind of mechanism aspects that the peptide molecules will be targeting. We found that the peptide which is stimulating BAX, caspase 3 and caspase 9 overexpressions are inducing that will leads to the inhibition of cancer cell mechanisms. So this is all about our research finding. So with this, keep a note on it. This NV14 peptide still under the laboratory level of research. It needs further in vivo model needs to be done, and some more aspects needs to be covered. So but it will be acting as one of the lead molecule as of. So we will be working on the other in vivo aspects about the NV14 peptide. So what will be the take home message? So peptides are the small molecules that possess various therapeutical advantages, like we can specifically design a disease targeted uh, peptide molecules as a therapeutic agents. Peptide drug discovery is the next generations of medication, and peptides specifically target the disease condition. So this is all about my presentations. If you want to read for further reading, you can go through these research article and our findings also. You may so I will be very glad to welcome all. You may go through and get follow my LinkedIn link. And if you are interested to know more about peptide based research or peptide therapeutics, or if you are willing to be collaborated, you are most welcome. You can mail, you can reach out me through the mail or phone number. Any questions? Welcome.
No, so thank you, sir. Thank you for the presentation. And I have some questions here.
Yes.
Yeah, okay. So, how will the integration of computational models and machine learning algorithms transform the landscape of peptide therapeutic discovery and development, particularly in predicting the efficacy and minimizing the adverse reactions?
Okay. So nowadays, before starting into the wet laboratory kind of thing, the computer-based or the bioinformaticians will be helping in the most significant therapeutic purposes. So molecular docking is one of that kind, such approaches that will specifically targeting our peptide structural configurations and our disease proteins configurations. So, based on the molecular docking approaches and simulations, and ADMET is the one of another algorithm which will be screening whether this peptide possesses to be drug or not. That can be visualized in silico tool itself. So apart from this, we will be having n number of prediction tools, which means we can able to predict its antioxidant potential, anti-inflammatory potential, antimicrobial, antiviral, anti-cancerous potential. So all these are all the different algorithm which will be working on what kind of amino acid combinations that we are going to predict or we are going to have the peptide sequences. So based on that particular amino acid combinations and its textural constituents, what kind of peptide linkage between one amino acid to the next amino acids, and what will be the size, what will be the molecular formula, what will be the net charge, and what will be the hydrophobic or hydrophilic and polarized natures of these peptide sequence. All will be we can analysis through a system based analysis. Then we will be further moving on this what it is various predictions of bioinformatic algorithm. Then we will be going further for what it is molecular docking and simulations. And this ADMET, it is a very good online platform which will helping the computer aided peptide based designing or peptide based drug discovery therapeutical approaches that will be helping to reach out to the in silico but it is computational into the wet lab kind of thing.
Yeah, okay, thank you, sir.
Yes.
Okay, as the role of the peptides in immunotherapy continues to expand, what breakthroughs are anticipated in utilizing the peptide therapeutics to modulate the immune system as more precisely and especially in the context of autoimmunity and cancer?
In the context of autoimmunity and cancer, we will be getting to know about what kind of molecule. For example, if we will be taking as a cancer, we will all know about it. What is cancer? Cancer is a uncontrolled growth of cells. So when it is happening due to the imbalanced state or uncontrolled condition or your for a P53 genes mutation due to some logical reason. If we specifically targeting or if we are able to regulate that improper function into be the proper, for example, getting mutation of P53 is stopped with the help of peptide, then it will inhibit the cancer proliferations. So in such a way, the ortho as well as the cancerous conditions, all the cancerous conditions will be having a different logical reasons and different proteomic targets to be approached. If with the help of the amino acid constitutions and with a known target of protein, what will be the disease condition? With tackling the amino acid combination of peptide, with thus that disease condition we can overcome and it will be used as a specific drugs in future.
Okay, so how might the advancements in peptide therapeutics contribute to overcoming the global challenge of antimicrobial resistance?
Antimicrobial, yes, of course, it is a very good question. Thanks for the question. The antimicrobial, so it is the most demanded thing as of, because why it is most demanded thing is the day by day the microorganism also evolving. Most of the drug which we are using right now, the as antibiotics, it is not killing the microbe because these microbes are getting evolved or it is getting adapted to that particular antibiotics. So it is acting as escaping or it is enriched with those antibiotics. So in that case, our peptide molecules, which will be more specific and it can kill the microbe in specific manner by degrading its plasma membrane or cellular membrane of those microbes. So peptide, n number of antimicrobial peptides are available and most of the peptide based drugs antibiotics are used in cosmetic industries, right?
Okay. Sir, what role will nanotechnology play in the future development of targeted peptide delivery system for cancer therapy?
Yes, of course. Because for the peptide based therapeutics, we require n number, which means the large amount of peptide molecule needs to be administered for therapeutic usage. So, in this particular case, if the route of to increase its bioavailability and biodistribution ratio, the nanoparticle based treatment like nano encapsulation or nano carrier based thing, as like I mentioned in the previous slides, that will help to deliver that particular targeted region. So it will increase the bioavailability of the peptide molecules. So this nano carrier are most of like liposome mediated, some most advanced techniques that will be utilized in the nano basis for cancer, peptide based drug deliveries in cancer therapeutics. But still, most of the research which will be in the peptide are developmental stages and it is in the preclinicals and clinicals. Very few, as of now, we will be getting 80 plus numbers of drugs as in peptide forms. Still, more number of peptide based drugs are in laboratory level and clinical levels. So it will be a future breakthrough by overcoming the nano-based or targeted drug delivery that will help n number of drugs that from peptide-based origins. So
Yeah, okay, thank you,
basically,
sir. Okay,
yes.
how can the peptide therapeutics be tailored to individual genetic profiles to enhance the treatment outcomes in personalized medicine?
in personalized medicine, like again it is going to be focused on, but each and individual, because we will be right now we will be having personalized medicine in, we will be screening out each individual's genetic profile based on the disease condition. So in that case we can specifically design what combination of amino acid will be given as a specified drug for that particular individual. That can be, this will be the approach on personalized medicine based on peptide therapeutics. We can specifically design based on individual's immune system, we can design a peptide based drugs accordingly.
Okay, okay, thank you, sir. This is my final question. So, how
Yeah.
will the future research address the immunogenicity and potential toxicity of peptide therapeutics in clinical applications?
Most of the peptides are considerably less toxic. If the peptide is originated from venom, or it is originated from some venom or some higher order proteins, that will be considerably slight some side effects. And comparing to the other phytochemical or secondary metabolites, this it is a proteomic in nature, so it won't have that much side effects. And comparatively, it will be, and it is very specific to the protein targeted regions. So that is why the most of the researchers are willing to work on peptide based drug discoveries in recent days.
Yeah, okay. So thank you, sir. Thank you for the answers. The answers are very useful. I hope the access and the participants can understand about that. So finally, as we draw this enlightening session to a close, it is both an honor and privilege to extend our heartfelt gratitude to Dr. Manikandan Velayutham for his invaluable contribution today. So, Dr. Velayutham, through your extensive expertise and insightful perspectives, you have not only enriched our understanding of peptide therapeutics, but also illuminated the path forward in unlocking the nature's vast potential for innovative and medical solutions. So, your dedication to the field of biotechnology is underscored by your significant academic and research achievements and serves as an inspiration to us all. So, the depth of your knowledge from the intricacies of drug discovery using the superfish models to the nuances of nanoparticle characterization and beyond has provided us with a comprehensive view of the challenges and opportunities that lie ahead in peptide therapeutics. So, moreover, your commitment to advancing the biotechnological research and education is coupled with your ability to mentor and inspire the next generation of scientists is truly commendable. So, your work exemplifies the profound the impact that dedicated researchers can have both on the scientific community and society at large. So, on behalf of everyone here, I extend our sincere thanks for sharing your time and insights and expertise with us. Your contribution today have not only sparked curiosity and fostered a deeper appreciation for the field of peptide therapeutics, but also encouraged us to continue exploring the frontiers of science and with a passion and perseverance. So, thank you, Dr. Velayutham, for being a beacon of inspiration, for playing a pivotal role in our ongoing quest to harness the potential of nature through the scientific innovation. We look forward to witnessing the continued impact of your work on the world of biotechnology and beyond. So, thank you, sir. Thank you so much.
Yes, thank you. Thank you very much for Tech Solutions for offering me to contributing my presentations through the sessions. Thank you very much to everyone. Thank you.
Yeah, thank you, sir. Thank you so much.
Yes.